Hello, and thank you for the invitation to present. It would have been a special pleasure to present in Montreal, my alma mater at the MNI, but alas, I'll greet you from home to your home. So today I'll talk about shaping the brain, and any of you who've worked in structural brain imaging will be quite familiar with a slide such as this one. In this case here, we're showing the changes in thickness of the cortex during normal development. And there's a normal trend line showing that in the posterior cortex, where this particular plot is pointing to, there's a decrease in thickness. But what's striking about this particular plot, and again, this is true whether you look at cortical thickness or brain volume or local change in shape or surface area of any particular part of the brain, is the variability. This is a normal population, and yet you see values ranging almost twofold in size. And it's this variance that's quite intriguing, which I'll try to focus on today to understand a bit of where does it come from, how do we explain all of this variation in shape of the brain, and does it matter? So first way to look at it is from human twin studies. So this here is an older study, and there have been more updated ones since then, looking at the decomposition of variants from twins, either monozygotic or dizygotic twins. And doing so, you can divide the variance into that's contributed by genetics on the left side, from shared environment in the middle, and from unique environment on the right. Now, any of you who've worked with these types of ACE models before will know that the unique environment encompasses everything that can't be captured by genetics and shared environments, so includes noise components as well. But still, the first take-home picture from looking at decomposition of variants in this fashion is that there's a dominant genetic component, there's a strong unique environment component, and a smaller but still significant shared environmental component as well. A more modern way to look at heritability, or another way of looking at the genetic component to brain structure, is looking at the imaging-derived phenotypes from the large biobank study recently published. And here you can see all those phenotypes ranging from volumes to surface area to cortical thickness, etc. And you can see the heritability on the y-axis, ranging from quite low values up to about 0.8, with the vast majority of imaging-derived phenotypes showing significant heritability, and again, averaging out somewhere between 0.3 and 0.6 for most of them. So I will address this topic by focusing on mouse research rather than human research. So of course, arguments for using mice are predominantly the superb genetic toolkit allowing us to genetically modify the animals and thereby gain precise insight into functions of genes. In addition, one of the reasons that makes this so useful is that there's significant genetic homology between humans and mice. And also, even though the brains are lissencephalic and of course much smaller, since they are derived from the same mammalian lineage, we can see similar brain structures from one species to the other. Of course, in addition to genetics, you also have tight experimental control over the environment. And in order to be able to use the same information, we can obtain very high quality MRI and image processing methods adapted from human brain imaging to give us very similar readouts. So I'm showing two examples on the right. We have an in vivo manganese-enhanced T1 image at about 60 micron isotropic that can be taken in about an hour and a half, and an ex vivo gadolinium-enhanced T2 weighted image at 40 micron isotropic, which is taken overnight in this case. So for those not too familiar with the mouse, again, the cortex out here is lissencephalic in this axial slice, in this coronal slice. You can see the hippocampus with the granular cell layer, the dentate gyrus, visible in both in vivo and ex vivo studies, as well as detailed lamination contrast in the cerebellum and other high detail, though of course it's not yet at the cellular level of resolution. But at this types of matrix sizes, it's not too far off from what you would get from high quality human T1 or T2 scans. This again allowing us to have information in the tightly controlled experimental settings of the mouse that's comparable to what we'd get in humans. So I will start with genetics. And in the mouse, the way that we would approach this is to take multiple genes, knock them out or modify them one at a time. And in almost all of these studies where I'll be describing single gene experiments, you will have a series of around 10 mutant animals, and then an equal number of litter-mate control wild types. Then of course, they're all imaged with one of these either in vivo or ex vivo structural MRI scans. We process the data to extract information about brain shape, local brain shape and volumes. And then we do the analysis, correct for multiple comparisons, exactly the way you would for human brain imaging. And of course, we repeat this from one gene to the next. And so the very first results I'll show in this regard is a large study we've been doing looking at autism-related mutations. And again, the focus here isn't on the autism part of it, but to use this as a set to begin to take some inference as to what kind of changes, in this case, in overall brain volume, can be induced by single gene mutations. So what you have here on the x-axis is a series of different mutations. And in each of these mouse models, we would have around 10 mutants, 10 letter mute controls, and then we'd compare them. The bars are in units of effect size, shown on the y-axis on the left, ranging from, in this case, what, -0.5 to 8. So in this case, the effect size is in units of standard deviation. So a -4 would be 4 standard deviations smaller than the letter mute controls. Also translated are, again, on the y-axis on the right-hand side in this case, from effect size into percent difference. And so a first look at this data shows that the majority of mutations have fairly low effect sizes, don't change the brain too much. And then there's a few that have significant effects changes on the brain itself, ranging above 10% or effect sizes of up to 8. So this is the same data where I've added now a marginal histogram, again showing this impact where there's most mutations have fairly little effect on the brain, and then a few isolated large ones are sitting at the effect size around 8, up to about 20% change in brain volume. So in this case, those few mutations that do show these large effects are almost all related to P10, which is known to be a macrocephaly gene. So here's one example of what this looks like if you have P10 or an unrelated but also macrocephaly mutation in NHS. You can see that the volume of wild types is in one study around 425 millimeters cubed, the other one around 450 millimeters cubed, and then a 50 to 75 millimeter cubed increase in brain volume in the mutants. And so the central story here is, now if I get rid of the direction and just look at the absolute effect size, is that most of these single mutations have significant effects on the brain, but fairly small in effect size. And then a few, which in humans would be considered macrocephaly genes, have a large striking effect above 10%. Now, this is not an unbiased sample. This is studies that was designed to look at autism and related genes. But it does make the point that in these mutations, often with disorders related to micro and macrocephaly, you have only a few mutations that have very strong effects, and effect sizes increasing above 10% difference. An alternate way to address issues of genetics in brain structure and size is by using polygenic mouse strains. So in this case, this would be recombinant inbred strains, where the core idea is that you take two inbred mouse lines, in this case, mouse line A and B, and then you start breeding them with each other for the first generation, have one more breeding of those particular offspring for a second generation, and then you inbreed these in turn, so this is sibling mating, until you get new inbred strains that share a unique combination of the two parental strains. So these two inbred strains will have differed in hundreds of thousands or millions of locations on the genome. And so there's a different share of that particular variation that's encompassed in multiple offspring that are in turn inbred again. So now this can be used for the sake of this talk as a model of polygenic variation. So it isn't a single gene that's been carefully controlled and knocked out or modified, but instead it's thousands or up to millions of variants where the strains will differ. So if I look at the brain volume in this case, you have a few of these strains, in this case from the DBX set of mouse lines, where the two founder strains are the DBAs over here, and the black sixes on this side, and then F2 crosses of them, again inbred in their own right, which share different amounts of genetic variation. And again, plotting this in the same way we did for the autism-related single gene mutations or small genomic region mutation, the effect sizes are much larger on average, where you get multiple effect sizes spanning above 10% volume or above an absolute effect size of 4 or 5. And so putting it all together, if I look at the single gene monogenic mutations, which are in red, and the polygenic variations, which are in blue, you see a distribution that's quite different, where the majority of polygenic variation causes high effect sizes, large percent differences between strains, whereas the single genes tend to have much smaller effect sizes unless it's the few very much related to macrocephaly type mutations. So a basic take-home story is not surprising to anybody who's followed the evolution of looking at variation in brain size, is that it's really additively caused by multiple genes, much more so than it is by single genes having a large effect. though there are exceptions, again, related to macrocephaly in this case. The related question then becomes, to what extent can we figure out what is happening in the brain if you knock out a single gene or if you modify a single gene by where that gene was expressed? So in order to answer that question, we took many of these individual models that I showed in the previous slides and aligned them to the Allen Institute Gene Expression Atlas. So what the Allen has done in a heroic effort over the years is look at in situ hybridization studies of all of the protein coding genes in the mouse genome and created maps of distribution of those genes across the brain. So an example is shown here, we're in GRAIL2, which is one of the sonic hedgehog genes known to pattern the cerebellum, where if you look at the MRI effects, shown in blue, you can see they're most striking in the cerebellum. And here in green from the Allen Institute is looking at where that particular gene in GRAIL2 is expressed, again, dominantly in the cerebellum. So in this case here, if you were to try to understand where the changes in brain shape would occur, knowing the gene expression, we'd come up with a fairly confident prediction. If we look across more of these models, so each of these individual dots here corresponds to a single mouse model, what you see is that that's a bit of a mixed story. On the x-axis here, you have the minimum false discovery rate, so basically the significance you obtain from the MRI part of the study. And on the y-axis, you have the maximum gene expression energy. Then every one of these dots is an individual mutation. In the circles, you have where there is preferential expression, which means there's a significant association with where the gene is expressed and where the changes occur. And in triangle, where there is no preferential expression. And then the positive predictive value is shown in the color scales, with the reds being the highest positive predictive value. So in those areas where you have a significant effect on brain volume and a decent level of gene expression, 8 out of 11 of those mutations have preferential gene expression. 3 of the 11 don't. But even then, if you look at some of these, the optimized positive predictive value can be as low as 0.2, 0.3, or 0.4, meaning that the actual ability to predict the changes is fairly low. An alternate way to look at this, in this somewhat complex slide, is not just looking at gene expression at the adult time point, which the previous slide showed, but looking at the gene expression of that gene across development. So here you have every one of these panels corresponds to a different mouse line, different mutation. And then on the x-axis, you have gene expression. Again, this is from the Allen Institute Developing Genome study. In embryonic day 11, 13, 15, 18, postnatal day 4, 14, 28, and then to adult time points. And so if you look at a few of these examples, what you can see is that the adult gene expression might not be highly predictive. On the y-axis is, again, positive predictive value. But some developmental time points might indeed be much more so. So in those cases, the way that you understand it is it doesn't matter as much where the gene is expressed in adulthood, but where during brain development the gene was expressed, and therefore then influenced overall outcomes at the end of the day. So as an interim summary from what I've talked about so far, is as we know from all the human studies and which motivated this series of studies to begin with, is that the brain's shape is highly variable, and that most variation can be explained by genetics, which is dominantly polygenic. So again, single genes contributing a small amount, adding up to a larger difference. Where the gene expresses only partly determines where the shape of the brain will change, more so in some genes than others. And often it is as important where the gene was expressed in development than where it is expressed in adulthood at the time of the imaging study itself. So switching track to environment, from the genetic component, one way to think of environmental contributions to brain shape is to look at studies of brain plasticity. Probably the first, or at least one of the earliest, very convincing of these studies in humans was the London taxi driver study by Eleanor Maguire and colleagues in 2000, where she showed that parts of the hippocampus were different in taxi drivers versus otherwise matched controls, and later also showed that that was more important for taxi drivers than for bus drivers, which are different ways of navigating around town. A later study in 2004 by Bogdan Rogansky and Arno Mai's group showed that teaching young adults how to juggle changed the posterior parts of the brain, where every 3 months interval, you get around a 3-4% increase in total gray matter, which after you ask them to stop juggling, decreased a bit. This was later replicated with some diffusion imaging data shown as well in Heidi Johansenberg's group in the work of Jan Schultz. And since then, there have been multiple studies looking at the effects of music training, of balance training, and so on, showing that the brain can indeed be malleable in adulthood at a scale that's detectable by structural MRI. So we wanted to look at this in mice in order to understand it. And so we created our mouse version of the London taxi driver study by training either bus mice or taxi mice, we jokingly called, on the Morris water maze. That's what you see in this video here. You see a mouse swimming. The pool has had white paint added to it, and the mouse is trying to find this platform. So mice are good swimmers, but they still prefer not to be forced to swim around. They prefer to find the escape from it. So this mouse in the first day is looking around. After 60 seconds, it's guided to the platform. And after a while, it learned exactly where the platform is and jumps to it. And then at the end of the experiment, usually 5 days, we can remove the platform and see if the mouse searches in the right spot, i.e. does retain a memory of where it was supposed to have gone. Now, there's two ways you can run this task, or two ways that we ran this task in. One of them is the spatial version of the task, or our taxi driving mice, where the mice use spatial cues located around the pool in order to find the platform. And the other one is a non-spatial cued version of the maze, where the mice just swim to a flag platform. So all they have to do is figure out where the platform is located, swim to it, but the location changes in each time. So if you use the spatial version of the task, which are these two rightmost indicators, you can see an increase in the volume of the hippocampus. Whereas if you use the non-spatial version, you can see an increase in volume of the striatum. So this gives us evidence supporting the evidence in the mouse supporting the human work that short periods of training, in this case only 5 days, allows changes in the brain that are detectable by MRI. So looking at what this looks like on a per voxel level, in red you have increases versus controls, in green you have, in blue you have decreases versus controls. You can see in the spatial version of the task you have extensive hippocampal changes, where you have only some hippocampal changes, especially focused on the dentate gyrus in the non-spatial version of the task, and then some increased striatal changes in the non-spatial version. Factoring out exercise as a potential confound here, you can see that in red areas where the spatial version is increasing volume compared to the non-spatial version after matching for exercise, you can see CA1 of the hippocampus has changed in volume bilaterally, and in the non-spatial version, you see changes in the right striatum and some of the right sensory motor cortex. Expanding out from this and looking at other tasks, another spatial version of this type of a task is the radial arm maze. Conceptually, it's quite similar, except that it's on dry land that the mice are rewarded with food rather than with an escape from water. And here too, we see very similar images that spatial training on the radial arm maze has these CA1 changes in the hippocampus in almost the same location we saw as the water maze, and decreases in this, also the right striatum, again, surprisingly lateralized. Training mice on a rotor rod, so this is a motor skill training where the mice sit on this rod, which is suspended a few centimeters above the ground, and the rod begins to rotate at increasing pace, and the mice have to keep up in order to not fall down. The fall isn't far, so it doesn't bother them much, but of course they prefer not to fall than falling. And if we do this for a few days, we can see again hippocampal changes, so in this case more in CA3 of the hippocampus, as well as some motor cortex changes and some striatal changes induced by rotor rod training. Switching tasks entirely and looking at a mix of sensory motor and pain, looking at the effects of tooth extraction done by colleagues of ours in dentistry under anesthesia and analgesia, we can see that extracting the rear molars changes the gustatory and insular cortex by decreasing it in volume, as well as some decreases in the pain system around the periductal gray, and some increases in some of the ascending midbrain systems in response. Another example to go through these series of studies shows, in this case, using a taste aversion task, that if you train mice for a while by exposing them to sugar, then after 14 days of that, condition them with an aversive stimulus that the sugar is unpleasant before allowing the memory to extinguish, you can see changes in the brain that correspond to each of these different stages, with decreases in volume around some of the gustatory and basal forebrain areas in sugar consumption, significant association with the insular cortex and again the basal forebrain and accumbens as taste aversion is applied, and then those changes seem to reverse as you go into the extinction of this taste aversion memory. Same thing is shown again in these plots here across these different time points. This was a longitudinal study, where if you look at the cingulate, for example, you can see that the water and sugar group match nicely until the taste aversion experiment part of it is applied. So you can see that here, where you have a jump if the sucrose was paired with aversive stimulus than if the water was. Whereas if you look at the fundus of the striatum, it's really an early effect of consumption that's different. Lastly, in this video, a more omnibus test is rather than training mice on any one individual task, simply putting them into this enriched environment that you see here, which has running wheels, it has mazes, it has more mice in it. And this enriched experience itself can then be tested to see whether that had any effect on the brain. But so a stronger stimulus than any individual task, but of course you can't as easily dissociate what it was that caused these changes. So in that case, again, you see significant hippocampal changes for putting mice into the enriched environment for a few weeks. Predominantly hippocampal with some cortical areas changing as well as some septum and some decreases in the cerebellum and thalamus. In this case, looking at the same study, you can begin to get a bit of a sense of timing by taking a few scans before you change the environment, and then putting the mice in the enriched environment and scanning them for a few times over 2 weeks, where the enriched mice are in the triangles and dashed line, and the standard housed mice are in the circles and solid line, where you can see that they begin to separate already after 24 hours, and then become significantly larger by around 4% by the time you fit 2 weeks. So just zooming in on this early time point, again, it's already quite visible by 48 hours and potentially even detectable by 24 hours, indicating that this type of plasticity in the brain is really quite rapid. So this was quite a whirlwind tour of different experiments. I didn't want to focus on any one of the plasticity experiments in particular, but instead I wanted to bring home the message if we're looking at what's shaping the brain. So this has been a whirlwind tour of different plasticity experiments, where the point wasn't to talk as much about the individual experiments, obviously not at that speed, but instead to drive home the point that the shape of the brain remains plastic into adulthood, that the effects are quite functionally specific, and that they're quite rapid. Some of these change detectable in a day or two, and then focusing on areas that you'd think would be implicated given the task, whether it's tooth extraction, whether it's taste aversion, or whether it's simple memory tasks. So what might cause these changes? There was a review from Robert Zatori and Heidi Johansen-Berg some years ago, which outlined different factors that could be contributing. So it could be neurogenesis, glial proliferation, angiogenesis, scale changes, dendritic branching, spinogenesis, or axonal sprouting. So again, there's a rich literature in cellular plasticity, and of course, in the mouse, you can begin to bring these two worlds together. So what we did is in that last enrichment study that I showed, in addition to the MRI, we also after 14 days of putting the mice in an enriched environment, we also looked at changes in gene expression in the hippocampus and in the visual cortex by extracting bits of brain and running bulk RNA-seq. And so the terms that are differentially, the genes that are differentially expressed, focus on, as you can see in this slide here, on the regulation of glutamine metabolism, exploration of behavior, social behavior. So this is the gene ontology enrichment study of those upregulated or downregulated genes based on enrichment, which we can then take back this rich information that you see here to the initial hypotheses generated by Zatori, Fields, and Johansen-Berg, and ask, what do we see evidence for in terms of changing genes after this enrichment, which did cause a volume change in MRI? And so if we do that, we can see that we do indeed have evidence for positive regulation of neurogenesis. We have evidence for the regulation of dendritic spine development and evidence for axonal development, though at least in this particular study, no evidence for gliogenesis and no evidence for angiogenesis. We can also take some information from timing. So here in gene expression studies, we're seeing the effects of dendritic spine development and neurogenesis, but they work on quite different timescales. So to take a shorter walk through plasticity or the timing of brain plasticity, within seconds, you have, after neurons fire responsive stimuli, you have long-term potentiation, which can last for minutes into hours. At that point, astrocytes swell, improving communication between synapses. New protein synthesis initiated after a few hours, and then in the period of many hours to a few days, you have dendritic and axon changes in size and shape. So a natural experiment that people have been using in order to understand this is to look at the estrus cycle in female mice. This was initially done in a few studies in rats and also replicated in mice, where across the estrus cycle, which lasts 4 to 5 days in mice and has 4 different stages, in proestrus, you have high estradiol, and that changes the number of spines that you have in the hippocampus. Then estradiol drops from proestrus to estrus, and the number of spines drops as well. It can also be seen by these gene expression changes, where P-ACT and TRAC-B, so the receptors for the AKT pathway and the BDNF pathways, are upregulated, as is postsynaptic CD95 and estradiol. Then they drop during estrus, and then begin to increase again as progesterone rises in diestrus. If we look at the volume of just the hippocampus in these studies, in these mice, so these are just normal black 6 female mice with changes, estrus cycle staged and volume changes measured. We can see that hippocampal volume is highest during proestrus, drops during estrus just as the spine changes do, and then begins to rise again mid-estrus and diestrus. So in this first study here, this was ex vivo with a large cohort of mice looking at different effects of staging, then replicating it in vivo by following a few mice across the estrus cycle, and again showing a drop from proestrus to estrus, and then increases again as progesterone and estradiol rise. So the plasticity studies show that indeed plasticity of the brain is rapid and functionally specific and measurable by MRI. And there's pretty strong evidence at this point using both the gene expression studies, some staining studies that I have not shown in this particular talk, as well as the timing information you can get from changes in the estrus cycle, as well as the early changes in the enriched environment, indicating that the likely dominant change, at least initially, is in neuropil remodeling, so changes in dendritic spines, synapses, as well as in axonal boutons, with predominantly later changes through neurogenesis likely playing a role, but not in the early time periods. But to bring back to this particular slide here, we're looking at genetic and shared environmental changes. I showed multiple changes in genetic influences on the brain, and I just showed multiple examples of shared environment, but how do they actually compare? So for that, we're going to do a meta-analysis of the different brain plasticity studies, so some of them I've shown in the previous slides, on its effect on mesoscopic brain plasticity. So in this case, we combined data from 16 studies performed in the labs, with a total N of about 1,000 mice, which were both male and female mice of various genotypes, but they were either trained on a maze, so the radial arm maze or the Morris-Water maze. They were given a simple exercise study, so they were given a running wheel, or the complex enriched environment that I showed before. We then take this data and do a hierarchical Bayesian model to determine the effects of maze training, exercise, and enrichment on hippocampal volume. And the way that we set up this study is that we do it in 3 levels of a hierarchy. The first is simply an active versus control. So do any of the interventions, whether it's maze training, exercise, or enrichment, have an effect of no intervention? Then can we separate out maze training versus exercise versus enrichment? And then lastly, what are the effects of the individual studies within each of these? So the very first level is any intervention, so active versus control. And what you see are the Bayesian credible intervals here, with the solid lines showing the 50% credible interval and the dashed lines, the 90% credible interval. So if you look at the effects of being active, so any of the types of intervention, is that we have an effect size of around 0.3, or total percent change in normalized volume, this is of the hippocampus, of about 1.5%. As we look at the different sub-studies, we can see that the effect scales with the type of intervention. With maze training giving a change of slightly less than 1%, or about 0.15 standard deviations, exercise about 1.5%, and enrichment a bit over 2% volume change, or a bit over 0.5 standard deviations. Looking at all of the individual studies, so these are, anything in green is a enrichment, is a maze study, anything in orange is an exercise study, anything in purple is a enrichment study. You can see that the maze studies vary, with some of them showing no effect or even a negative effect, and some of them showing positive effects. Whereas all of the exercise studies and all of the enrichment studies show a strong positive effect. Another way to think about this, if we switch back to frequentist statistics, on the maze studies, about 11 of 17 would have found a significant effect on the hippocampus, whereas on the exercise or the enrichment studies, all of them would have found a significant effect on the hippocampus. But still, it's the absolute units that I want you to pay attention to here. We're seeing very small effects. So this is a volume change of less than 1% in many cases, or certainly less than 4 or 5% in almost all cases, except for one study, which I'll get back to a bit later. So bringing it back to an interim summary is that we have strong genetic changes, they're predominantly polygenic, and the effect sizes we saw of that were often exceeding 10%, getting up to 20% changes in brain volume in the large macrocephaly genes, or in many of the polygenic variations from the recombinant inbred lines. For the environmental manipulation, so these are adult changes due to brain plasticity, we see significant effects that are functionally specific and that are quite rapid, that are likely explained by neural modeling, but that are small. So 1% to 3%, which of course can be detected using the small variation you have in tightly controlled mouse experiments, but are more of a challenge in human studies. We're trying to keep variation under control. It's often quite difficult. So I'm going to get back to that enrichment. You might remember the last slide of the different effect sizes had one particular study that was much larger in effect, and that one was actually a neonatal enrichment study. So you've seen this before. If you enrich mice, so you put them into this cage with extra running wheels and mazes, etc., you can see these changes that are distributed throughout the hippocampus with some cortical and cerebellar effects as well. If you place my dams into an enriched environment a few days before they give birth and then follow the pups as they're raised in the enriched environment, you can see that the hippocampal changes are much, much more profound. So this is where we get the 7% change in volume as opposed to the 3-4% that you get from adult enrichment. But also qualitatively somewhat different. Not only you have much more hippocampal change, you also have some cortical surprising decreases, likely due to changes in synaptic pruning that you don't see at all if you look at the adult enrichment studies. So some evidence that not just the type of plasticity change makes a difference, but when you do it has a huge impact. We're early showing bigger intervention. This is at home in some of the more medical type studies as well. So in this study from my colleague and friend Brian Nyman, he looked at the effects of brain irradiation on mouse development. So the point of these studies is to look at irradiation as an effect of oncology studies, where often irradiation for brain tumors is part of the treatment course, which is known to have its own effects that are detrimental to the brain, likely due to killing not just dividing cancer cells, but also dividing neurons. So thereby particularly affecting the olfactory bulb in the hippocampus, as well as in white matter areas. Now they did multiple studies over the years, and this one they summarized looking at the irradiation at different time points. So what you have here is the controls always sitting in red, and then irradiated cohorts of mice with the mean effect shown in the dots and the fitted line and the dashed line, whether they're irradiated at day 16. So this would be early childhood in comparison to humans. At day 18 or 20, by day 29, you're hitting around puberty, and then post-puberty at day 36 of adolescence. You can clearly see that there's an age-dependent effect. In this case, it's the fornix. In this case, it's the dentate gyrus from two different areas with the same pattern, where the earlier the intervention occurs, the more significant the effect is on adult brain shape. Somewhat similar, we looked at a study of rapamycin, effects of rapamycin on the brain. So this is rapamycin targets the mTOR pathway, which can be considered likely treatment candidate for potentially variants of autism, particularly for tuberous sclerosis. In previous studies, they've shown that rapamycin treatment can rescue social approach and familiarity deficits in TSC1 mutant mice, but they also induce volume decreases just due to treatment alone. So we examined this in wild-type mice, looking at the effects of rapamycin treatment at these therapeutic doses. And what you see here is in blue, this is over 4 weeks of treatment followed by absence of treatment to see if there's any recovery. In blue, you have vehicle treatments with saline, and in red, you have rapamycin treatment. In the solid line, you have treatment starting at week 6. In the dashed line, you have treatment starting at week 10. And so what you can see quite strongly is that there's significant decline in brain volume of around 7-8% with rapamycin treatment at week 6, but only about 3-4% decrease in brain volume if treated at week 10. And this is just where the effect occurs at the week 6 time point, showing that it's predominantly cortical and striatal effect that you see. So again, evidence that the effect on brain structure is dependent on when the intervention begins. So we saw that with enrichment, we saw that with the radiation, we see that with this particular drug treatment. Similar in an earlier study by Anne Wheeler, she looked at the effects of cocaine on brain shape. So in this case, mice were injected with 20 mg per kg of cocaine or saline for 20 days. And she again had 2 cohorts, starting in adolescence, so just before puberty or around puberty at 27, or in young adults at postnatal day 60. In the adults, there were no significant effects that we detected due to cocaine exposure at all, same 20 days, same dose. Whereas in adolescents, there were significant effects on the brain, distributed across the orbital frontal cortex, striatum, accumbens, anterior cingulate cortex, many of the areas you might expect would be involved in cocaine and similar types of sensitization. In a further example, in terms of treatment effects, we can look at a genetic model of rescue of Rett syndrome. So in this example here, this is a replication of a study that was done by Adrian Bird's group in Edinburgh, where they looked at the reversal of neurological defects in a mouse model of Rett syndrome, so Rett syndrome being caused by MECP2. And they showed that motor behaviors were recovered and mice lived longer. And so the way that this model works is that the mouse has this particular stop codon inserted between exon 2 and 3 of the MECP2 gene, but it's surrounded by two sites, two LOXP sites, that can be cut out at a time of our choosing. So effectively, these mice are bred with Cre-ER mice. Upon the injection of tamoxifen, it binds to Cre-ER, which in turn excises the stop codon, causing the gene to be expressed normally again and have functioning MECP2. So this can act as a model of treatment, where you can say that this obviously couldn't work in humans because the genes aren't engineered this way, but in the mouse you can reintroduce gene function at the time of your choosing to see what the effect of timing of gene production has on brain deficits. So I'll just show the overall brain volume deficits in this particular effect here from the MECP2 rescue study. So in red, you have the functional mice. So these were always MECP2 fully expressed, always normal wild-type litter controls. And then in blue, you have the completely silent mice. So these are MECP2 silent throughout. And in green, you have different timings of rescue. So looking at just the males, age is on the x-axis, where if you look at normal brain development between age day 50 and 100, you can see that it's still a slight amount of growth that occurs during this period of time. At baseline, so this is just when symptoms begin to appear, there's already a significant reduction in brain volume. If you leave MECP2 silent, you can see that the loss of brain volume continues over time. If you rescue MECP2 at day 50, you can see that brain volume stabilizes and recovers a little. If you rescue at day 80, it stabilizes and recovers then. So again, the effect that you have seems to be quite dependent on when you induce rescue. This is even more noticeable in females. So this is an X-linked gene, so the expression in females is much milder. Where at day 200, you still have a small amount of growth, negligible though over in wild-type mice, but in either the silent or the rescued mice, you see no changes in brain volume anymore. So again, more evidence that changing brain volume in terms of environmental interventions needs to occur early in order for it to have a sizable effect. We can look at what really begins to happen in the early stages using longitudinal MRI. So these are methods developed by Camilla Schultz-Lerch, where you can scan mice in vivo, starting at postnatal day 1, going every second day or slightly longer periods if you so choose, to look at the effects on the brain. So early development is quite rapid. So whole brain volume goes from about just over 100 millimeters cubed at postnatal day 1 to close to 400 by postnatal day 11, with all parts of the brain growing at fairly rapid clip. One way to look at this data, though, is to also begin to try to see where some of this variation begins to occur. So here, what this particular figure shows is different ways of fitting the developmental data. And what you have, if you look at this example over here, you have 3 different lines. In the gray line, you have the population effect. So just what's the mean effect of development in volume in this particular point. In blue, you have if you allow for a separate intercept per mouse. So you allow the mice to be different in size, but to assume the same trajectory. And in green, if you allow separate trajectories as well. What map A shows is where the allowing a separate trajectory per mouse gives a significantly better fit than assuming that the development trajectories are the same. And you see a lot of the classic hotspots due to early environmental interventions come up with changes in sensitivity to individual trajectories, especially strong in the hippocampus, in the VTA, around the locus coeruleus, in the cerebellum, and some of the midbrain structures. So effectively, what this is beginning to get at is not just the overall environmental effect, but the unique environmental effects that are hard to capture otherwise. Another way to think about it is to take a similar study, in this case from a follow-up study looking at sex differences in the brain, and ask questions about anatomy prediction. So can we take the adult time points, so either P65 or P36, and predict the volume for every mouse using the earlier developmental time points that were also imaged? And so the basic question, if you look at the observed in the solid lines and the predicted in the dashed lines, answers that we can very well. So this is just 2 examples from the primary motor cortex and the striatum, where the solid line would be if you predicted just using the population average, this dashed line here that goes across, and then the individual mice are shown as individual bars, where clearly the predictions are quite close to the actual observed values. This is showing that the mouse brain begins to be fairly stable by using developmental data to predict adult time points. But we can also see when this begins to emerge. So in these plots here, what you have is the predicted from own time points in the dashed line, and the distribution if you try to predict one mouse using the data from the other mice instead, showing in the gray line. So again, overall, the x-axis is the root mean squared distance, so an error measure, so the lower the better. Overall, using the mouse's own data is quite a bit stronger than using the other data. If we move along across different time, you can see that that begins to emerge over time. So using only the day 3 postnatal scan to predict the day 36 adult scan gives you relatively poor performance, whereas by the time you get to P17, using the prior data begins to give you quite strong performance. And interestingly, though somewhat tangential for this particular talk, that's particularly stronger earlier in males, where you can use earlier data to predict adult time than in females, where you need to include some more later data to predict adult brain volume. Lastly, on this line, you can look at exactly how environmental manipulation affects the brain by manipulating it as well. So we showed earlier that early environmental enrichment changes the brain significantly. But what's interesting about it is that in mice, they're born somewhat precocious compared to humans, and they actually don't open their eyes for the first days. So the question then becomes is how early do the interventions make a difference? And if so, what is it that's causing environmental enrichment to have an effect? And then a likely explanation of this could be to look at the effects of maternal care, how environment changes maternal care and how that influences brain development in turn. So we set up the study where dams were placed either into an enriched environment or into a standard environment, and then they gave birth to 2 litters. And sometimes they switched between litter 1 and 2. So in green, you have mice that were in the enriched environment for the first litter, in the standard environment for the second litter, in blue the other way around, and in red and purple where they stayed in the same environment across the 2 litters. This allows us to isolate the effects of what indeed does housing have on both behavior and brain. So looking at different measures of maternal contact, so how much time they spent with their pups, we can see that the amount of maternal contact in enriched environments is quite a bit higher. So it's 68% in enriched environments versus 46% in standard housing. And that indeed switches around, where mice that start off in the enriched environment, in the standard housing, end up having more maternal contact in the second time when they move to enriched housing. Whereas mice that start in enriched housing have higher contact and then much less contact in the second time around if they move to standard housing. And the same is true for nursing and multiple other measures of maternal contact. And brain volume across these 2 litters follows the same pattern. In multiple areas, so if we look at here, the accumbens as an example, where the volume is largest in the mice that are in standard housing, which stays constant if they stay in standard housing, or if they start in enriched housing, the volume is smaller, which stays constant if they stay in enriched housing, or it crosses over if you switch from enriched to standard housing across the 2 litters. And we see the opposite pattern, so it's increases in the habenula, same in the parietal gray, and same in the medulla. So ultimately, what you seem to have is early environmental changes where certain parts of the brain are susceptible to those changes, likely mediated by differences in maternal behavior or partly mediated by differences in maternal behavior that are caused by the changes in the environment that the dam finds herself in as she's raising her pups. So to summarize these particular studies, we have plastic interventions that seem to make a significant difference in terms of shaping the brain, but are quite subtle if the effects are done in adulthood, but are quite strong if the effects are done early. We suddenly get effect sizes around 7-8% or larger. Depending on how you study this, you can think of that as a shared environment if you can isolate the individual components, or especially later when you get to uniqueness in maternal care, which begins to also separate by environment, as well as uniqueness in terms of susceptibility to early environment, even mice raised in the same housing can get at the unique environmental components of brain shape. So I'll finish up by briefly touching on structural covariance, where we know that the brain doesn't change just in isolated brain areas, but instead changes in networks. So some of the early studies showed that if you look at across a population, the thickness of the cortex in Broca's area, it correlates quite strongly with the thickness of cortex in other language areas such as Wernicke's. So effectively what's saying is that if you have a larger Broca's cortex, you're also likely to have a larger Wernicke's cortex. Now there's different ways you can think about what might have caused this. You can think of it as an effect of connectivity. So the activation of region A causes synaptic remodeling of region A and growth in region A. which is also connected to region B, causes related growth between the two. Or you can think of it as an idea related to gene expression, where there's different developmental cues that happen to be shared in those areas, causing a correlation of change. So we looked at these studies using a series of different relations of measures between MRI and the Allen Institute, as I alluded to at the very beginning of this talk. We either looked at connectivity using the Allen's tracer studies. So these are retrograde injections of viral tracers, transcriptomics, so genes that are expressed similarly, or a simple distance function. Errors close to each other are more likely to be similar to each other. And so if we model that all, we can see that if we include all components all the way over here, distance and gene, as well as polysynaptic contacts, we can explain close to 40% of the variation in covariance using these different measures. Any one measure explains only around 10-15 of these particular points, but with two measures, you can get close to that by looking at just a combination of distance and genes, with also similar but slightly less effects of distance and monosynaptic connections. So effectively, what this seems to suggest is that everything matters. So you can get both an effect of distance, so the closer areas seem to correlate with each other. If they express the same genes, they seem to be related to each other, and if they're directly physically connected. We can test this last hypothesis by actually looking at a particular set of mouse models where there is some disconnectivity. So in this case, a genesis of the corpus callosum. An example here we'll use is the BTBR. So you can see these diffusion images showing the normal corpus callosum, black sixes, or FEBs, two different inbred strains that have a normal corpus callosum, or the BTBRs, which clearly do not. And so we again used the interhemispheric atlases of the Allen Institute and looked at changes whether we relate the seed ROI with either its ipsilateral connection or its contralateral connection, and ask whether that particular connection crosses a track that's missing in the BTBR mouse or that's present in the BTBR mouse. So this is 131 total sets of IPSI contra regions of interest that these studies were done in. And so if you look at the results, which you see over here, is the structural covariance tends to be higher in ipsilateral than contralateral. And in red, you have the mice that have a crossing corpus callosum. In blue, in mice that do not have a crossing corpus callosum. And you can see that the effects are no different between the two. So it seems to suggest that what's really driving this kind of covariance in terms of brain structure development is not just the fact that the two areas are connected. It seems to have much less of a role because you can test in this when you've disconnected the mice, but instead seems to be a correlated developmental patterns, correlated developmental pathways that seem to dominantly drive covariance between the two. So to finish up this talk, I started with talking about the high variability of brain shape and with its significant genetic components, which using the mouse as well as human studies, we can show is dominantly polygenic. And where the change occurs can be partly determined by where the gene is expressed. If not particularly strongly, the predictive power isn't great. And often it matters as much where the gene was expressed in development as where it's expressed in adulthood. There is definitely an environmental component as well, where brain shape remains plastic into adulthood. We can induce brain changes using different environmental manipulations, which are quite rapid and functionally specific. And they seem to be explained by neuropil modeling, as we showed using the gene expression data, as well as some of the inference from timing of these events. But they're quite small effect sizes, and depending on the type of study, between 1% to 3%. The brain is much more malleable in early development. So if you take some of the same interventions and move them early, and we can see this across different medical interventions, such as radiation or drug treatment or gene reactivation, where if you do it earlier, the shape of the brain is much more susceptible to identifying what happened than if you try to do it late into adulthood. Lastly, the brain shape is coordinated along networks, not just changing in isolated places, which at least in normal variation seems to be dominantly explained by coordinated gene expression as well as proximity, where direct connectivity actually matters less than we would have thought. So I'll finish here. I will thank many people, especially my wonderful lab over the years in Toronto, as well as my new colleagues at the Wellcome Center for Integrative Neuroimaging in Oxford, and the years of inspiration I've had from close friends and collaborators, especially starting with Alan Evans and Alex Seidenboss at McGill, and including others such as Lauren Arman, who've been wonderful collaborators over the years. And with that, I'd like to thank you for your attention.